Our prompt is how anthropomorphizing animals in films like Ratatouille creates bias against animal research. To start things off, let's meet Remy from Pixar's Ratatouille.
Ah, Paris, France. Home of the finest restaurants and the greatest chefs in the world. All my life, I've wanted to be one of them. You may think that's a strange dream for a rat, but I've always believed that with hard work and a little luck, it's only a matter of time before I'm discovered.
So, what exactly makes him anthropomorphized? Let's take a look. Anthropomorphism is the attribution of human characteristics, including emotional states, physical anatomy, or behaviors, to non humans. In this case, an anthropomorphic character is giving human actions and behaviors to an animal. In this example, Remy is anthropomorphized by standing on two legs and washing his hands. In the wild, rats are found on all fours and do not wash their hands. The history of animals have been used in the film industry since the 1920s and has been popularized by animation to bring about characters like Mickey Mouse and Bambi to the big screen. The film industry continues to anthropomorphize animals in movies like Zootopia and B-Movie. However, screen time for animals appear to be biased in species. David Pearson analyzes that the majority of the Discovery Channel's nature programming consists of animals that fall in the cuteness or repulsive dichotomy. For example, there are more programs about cats and dogs than pigs. Animal experimentation dates back around 300 BCE in ancient Greece. The goal of animal research is to use a genetically and physiologically related model to study the effects of drugs, diseases, and other conditions and see how that parallels to humans. Essentially, it allows us to experiment and learn without the use of a human subject. Some common lab animals include mice, rabbits, zebrafish, fruit flies, dogs, and non-human primates. However, there is some controversy towards animal research. Animal rights groups believe that the use of animals as experimental subjects is immoral and inhumane, and argue that some research can induce pain and other negative health outcomes towards animal subjects. Suggested alternative methods include stem cell research and computer simulations to prevent animal experimentation. However, it should be noted that most animal rights protests tend to be selective in which animals to stand up for. As an example, there are more animal rights protests towards experimentation with dogs than with fruit flies.
Next, we'll be discussing the neurobiology behind our response to animals in films. Empathy is key to this discussion, and various processing methods, brain structures, and chemical signals influence how viewers react to films. From emotional soundtracks to close camera shots, films are framed in a way to elicit the largest reaction from viewers. One way that our brains process these movies is with top-down modulation. In contrast with bottom-up modulation that processes sensory information, top-down modulation involves the active process of adding context to what we see from our past experiences or knowledge. With the context clue of how animals wear clothing and stand on 2 feet, top-down modulation allows us to interpret and accept this anthropomorphism of characters like Remy. Next, mirror neurons are a type of brain cell that respond equally when we perform an action and when we witness someone else perform that same action. This effect can be seen in the image where the monkey's brain responds similarly to picking up the banana and seeing someone else pick up the banana. This connects to the concept of empathy and shows how through mirror neurons, films can put us in the character's shoes to elicit emotional responses. Moving to brain structures now, many of the structures involved with the processing of emotions in social situations are implicated in how viewers respond to movies. The first region of interest is the prefrontal cortex, which is associated with logic, deliberative analysis, and self-awareness. Specific regions such as the ventromedial prefrontal cortex and orbital frontal cortex are implicated in assigning social and emotional meaning to our surroundings. One research group noted that introspective regions involved with rational processing, such as the prefrontal cortex, are inhibited during the sensory motor processing of watching a movie on the screen. By inhibiting these regions, viewers are able to really lose themselves while watching the movie. This lends to our acceptance of the anthropomorphism of characters like Remy, as viewers are able to suspend their potential disbeliefs that a rat could cook. The next region is the temporoparietal junction that is involved in the mirror neuron system and is important for mentalizing experiences of others. It is also implicated in the self versus other distinction that is crucial for empathy. One experiment that demonstrated this was using transdirect cranial stimulation on the right temporoparietal junction in theory of mind and cognitive empathy tasks. The theory of mind is the ability to attribute mental states such as beliefs, intents, and emotions to oneself and to others. When inferring another's intents or emotions, those that receive cathodal or inhibitory stimulation showed a significant decrease in accuracy in interpreting theory of mind and empathy tasks. This represents that the right temporoparietal junction is crucial for the processing and application of empathy. The final brain structure discussed here will be the right supramarginal gyrus that leads to the egocentricity bias that allows for maintaining the self versus other divide. One study performed paired participants together and both were exposed to either a pleasant or unpleasant simultaneous visual and tactile stimulus. For example, while participant 1 may have seen a picture of maggots, participant 2 saw a picture of a puppy. The two participants are then asked to evaluate their own emotions and their partner's emotions. If both participants were exposed to the same kind of positive or negative stimuli, they found it easy to assess their partner's emotions. But if the two participants received different kinds of stimuli, those that experienced unpleasant stimuli assessed their partner's good experiences less positively. These differences were amplified with the stimulation of the right supramarginal gyrus. This study suggests the importance of the supramarginal gyrus in the perception of empathy. By recognizing its importance and how individuals tend to be egotistic when viewing others, films can take advantage of this by presenting scenes that encourage the viewer to empathize with the character. Moving on from brain structures, various hormones and neurotransmitters are implicated in our response to films. Dopamine is a neurotransmitter and hormone implicated in reward pathways and emotional salience. For example, dopamine D1 receptors are upregulated in mated prairie voles that practice monogamous mating habits and have high partner preference and social contact. It has been shown to be released in higher concentrations when watching TV shows and films. Oxytocin is another neurotransmitter in the brain that regulates social interaction and sexual reproduction. Empathy is highly correlated with a spike in oxytocin levels. This is important for facilitating empathy when watching films because our brains are not perfect in distinguishing between actual human beings and those that we see on the screen. This effect can be even extended to animals that appear to have human characteristics through anthropomorphism. Finally, serotonin is a hormone implicated with mental alertness, better mental health, and decreased sensitivity to pain. A study found that blood pressure levels decreased and serotonin levels increased when interacting with a human's own dog, but not with unfamiliar dogs. This can be translated to our responses to animals in films that over the course of their narrative we grow in an attachment to and connection to. Various aspects of our neurobiology contribute to empathizing with animals on screen that we can then convert into a bias towards potentially harming animals through research.
So what biases result from anthropomorphizing animals in movies? The Bambi effect. The Bambi effect, inspired by Disney's 1942 film, is known as an objection to the harm of animals that are deemed to be cute. When a rat like Remy is anthropomorphized and made to look cuter than a normal rat, the audience doesn't want anything to hurt Remy or his adorable rodent relatives. Animal research has the potential to hurt Remy, thus the public may be biased against it after watching a film like Rataouille. The Bambi effect is extremely relevant to society. The infamous death of Bambi's mother, despite taking place in 1942, still resonates today with children and adults of all ages. In the year following the film's release, hunting rates were reported to have fallen by 50%. In addition, multiple proposals to reduce overpopulated deer herds were shut down due to passionate public opposition. Films like Rataouille may generate sympathy for rats and mice that are used for research purposes. Similar to what happened with the overpopulated deer herds, the public may come together to protest and has the potential to cause change and reductions in animal research. Protests against animal research can be found right here in Westwood. This Daily Bruin article, published April 27, 2008, details a recent march by animal rights groups around campus to protest the use of non-human primates in experiments conducted by UCLA scientists. One UCLA professor that the animal rights groups were marching against is Dr. Michelle Basso, who uses monkeys to study Parkinson's disease. Protesters believe that Basso's use of animals is unnecessary and that other technology can reach the same goals. Dr. Basso, however, argues that technology to replace animal research is not yet ready and that only the most impactful proposals involving animals are provided funding. She also assures that, quote, "Our animals receive better health care than many people here in Los Angeles." So what can we learn from these biases and how can we combat them? Biases against animal research can remind us of the importance of empathy and humanity when we pursue our own research interests. To combat these biases, we can attempt to improve existing policies regarding animal research. One current U.S. law is called the Animal Welfare Act, or AWA. The AWA, passed in 1966, regulates the handling of animals and requires licensing to prevent illegal pet theft and sale to research facilities. Despite being passed to protect animals, the AWA does not cover the most common lab animals, which are mice, rats, and birds. Despite being passed over 50 years ago, the AWA still does not protect these animals. In fact, a congressional amendment passed in 2002 permanently excluded these animals from the AWA. Another federal standard, the Public Health Service Policy, does provide protection for all vertebrate animals used in research, but only applies to PHS-funded experiments. In addition, accreditation for animal research facilities is done only on a voluntary basis. The 3 R's are a set of ethics that are a common part of animal research culture today. The first R, replacement, refers to methods that avoid or replace the use of animals with inanimate systems or animals that are scientifically proven to have lower pain perception. The second R, reduction, aims to decrease the overall number of animals used or to maximize the amount of information obtained from each animal. The third R, refinement, seeks to modify procedures in order to minimize pain and distress while enhancing animal welfare. One way to improve current regulations and combat animal research biases is to look to foreign policies in animal research. Several European countries are known to have some of the strictest regulations. In France, for example, the use of large monkeys is forbidden and painful tests must be done under anesthesia. All facilities must also be authorized to perform animal research. To become accredited, buildings must follow a strict code of standards for animal living conditions and employees must demonstrate their knowledge of proper animal handling. This is done every 6 years to ensure a continual high standard of care. These policies provide protection for all vertebrate animals as well.
In conclusion, the film's industry's use of anthropomorphization has indirectly created biases in selecting animals for future presentations. Films are able to use the neurobiological pathways implicated in empathy to encourage viewers to empathize with animals such as Remy from Ratatouille. And last but not least, biases against animal research can be combated by improving current regulations to be more empathetic towards animals. Thank you.